So we'll be doing a breakout session on these two topics. And the other people on the panel are Neil Davies, Katie Barker, Michelle Barbier. I've invited Michelle and Ariana to also sort of tag team on this proposal because the one of them knows a lot more than I do about access and benefit sharing, and Ariana will talk just for a few seconds about the agreement. The Memorandum of Understanding is one word for it in English. The sort of what kind of institutional agreements do we need in order to make Ocean Sampling Day actually happen. So bioarchiving, we've been doing it for a long time, as I said, in museums, and I put some pictures at the bottom just because I like to look at pictures of collections. But when we think about these things, some of it will be objects like this. Up above, you see what happens if you bioarchive for a couple of hundred years. This is just the collections in the Smithsonian, about 2 million georeferenced things. There's about 3 billion specimens in museums and botanical gardens around the world, so that's where we're headed. And I already showed this, but as I said, we've ponied up about 10,000 2 mil slots for Ocean Sampling Day. So the breakout session today could be talking about a number of things. One of them is, what are we going to put in these tubes, and how should we treat those samples when you do it? For example, one of the things they'll be doing is taking about 5 liters of ocean water, spinning it down through a filter paper that is contained inside a styrene plastic container that's sealed on both ends with clay. I think that what I would suggest is that either make 2 filters, or you actually crack that open, tear the filter in half, give half the filter to the bioarchive, and then you do science with the other half. Now, that, of course, could dramatically affect your workflows. Some of you may not want to do that because there's the possibility of contamination of the filter paper, but it gets rapidly into sort of nitty-gritty issues about what you want to do. And if you do want to send us samples, or you do want to preserve or archive some of the samples that you're thinking about collecting during Ocean Sampling Day, we really need to talk about how it happens. For example, that might involve a workflow going from the actual site to Argonne National Labs. It's going to be doing some of the work, and then they have to transfer it to us. So now, I don't know if I'm actually supposed to talk about this or not, but John Deck, are you going to deal with things like the informatics of associating samples with data? Where'd John go?
I'm up here.
Yeah.
We may not get down to that
kind of detail. Well, I'll just say that as everyone here has been talking about tying these data sets together, relatively few of you have addressed how you actually link data to a voucher specimen, which is the sample itself, or the phenotype if you're doing genomics. So there is one group of people who are doing it, though, and they're collaborating with our international initiative, and that's the folks in Berlin have actually linked up 6 different bioarchiving places. The reason why it's hard to do is because of this, that there is no agreed upon set of terms that describe tissues or the kinds of samples that you're going to collect in such a way that you can machine reason across those or do what John wants to do. There is one standard, and that is from the ABCD DNA standard, and you see it's the DNA extension for ABCD, but ABCD is one of the classical data standards for museum collections, but it's the rarest in the world. The one that most people use is something called Darwin Core, and Darwin Core doesn't have that, so it's really a very necessary thing to figure out the genetic extension of Darwin Core in order to get any of this off the ground. And just to explain why it's a hassle is that at the bottom you see what are represented as information systems for museums. The problem is that freezers have to have their own software, in order to be able to keep track of where the tubes are and what the metadata for those tubes are. But where we're headed, at least in the GGBN, is towards a semantic data aggregation layer, so that you really don't need to worry too much about the back-end details of what the IT is doing, but instead exposing the data to users. So another issue is what is a genome quality sample? How are you going to? This is about the only paper that I know of. It just came out in GigaScience earlier this year, and it's talking about tissue sampling methods and standards for vertebrates, for things like prokaryotes and other things. That's quite a different way of knowing. In other words, once you put the sample in the biorepository and someone wants to come back 10 years and work on it, one of the things they want to know is what is the quality of that sample? What am I likely to get out of it once I look at it? Their suggestion is to come up with a 4-star system for the quality of the sample, which I think this is actually a little whacked because in vertebrates, they're actually looking for 1 milligram of DNA in order to do, for example, the 10,000 genome project for vertebrates. But you can imagine that if 1 milligram of DNA, let's say, is a 4-star level, fulfillment of that particular criterion. If you go over to, say, specimen type, you might have 1 milligram of DNA, but it might be a sort of a 1-star kind of specimen. In other words, it's a voucher that you subsequently sampled. It wasn't actually live caught or freshly euthanized, and the storage also could be different. So over and above keeping track of what we have, we also want to have some kind of, I think you're going to want to have some kind of quality standards for knowing what that sample is likely to yield. So with that, I think, this is just to point out that the genomic community has been really quite bad at vouchering, anything. A lot of things in GenBank have no physical vouchers. There's no phenotype, so you cannot actually tell whether or not something where the DNA came from. And a lot of the reference genomes, my favorite one actually as an example is the cow, or the dog, it turned out, was Craig Venter's pet boxer. And Craig Venter's pet boxer is still alive, and there's reason why you might want to actually get the dog. So Jim Watson actually might donate himself. Anyway, now I'd like to introduce Michelle Bardier, who's going to talk a little bit about the access and benefits sharing thing, which is it's a part of the Convention on Biological Diversity, and it's a law, and anything that we move across international boundaries, as well as it's a good thing to do. And then after that, I'm just going to set up that Ariana will talk about the model agreement, a draft model agreement. So if you're interested in actually participating in Ocean Sampling Day, you need to think about that as well. Thanks.